Hello, people. Ever wondered how your body creates urine from the blood? Let's talk about the nephron, the microscopic unit inside your kidney that takes a simple filtrate of the blood and modifies it into the urine. Many changes takes place in the different parts of the nephron before true urine is created for the disposal. So from this point forward, we will call it forming urine, describing the filtrate as it changes along the way. Now, what's the principal task of the nephron population? To balance the plasma to the homeostatic set points and excrete the potential toxins in the urine. They achieve these by performing three main functions. One is filtration, second one is reabsorption, and the third one is secretion. But they do even more. Nephrons also have secondary functions that exerts control in three important areas, that is blood pressure regulation through the production of the renin, and the red blood cell production via the hormone erythropoietin, and the calcium absorption by converting calcidiol into the calcitriol, the active form of the vitamin D. So, yes, the nephron isn't just making a urine, it's literally helping regulate blood pressure, oxygen levels, and your bones. We are now going to see the structure of the renal corpuscle where urine formation begins. Now let's zoom into the very beginning of the urine formation, the renal corpuscle. As discussed earlier, the renal corpuscle contains of the tuft of capillaries called the glomerulus that is largely surrounded by Bowman's capsule, also known as an glomerular capsule. The glomerulus is an high pressure capillary bed nestled between two arterioles. The efferent arteriole bringing a blood in and the efferent arteriole taking it away. Bowman's capsule surrounds the glomerulus to form a lumen, a space that captures and directs the filtrate into the proximal convoluted tubule. We are now going to see the podocytes and the filtration slit system. Bowman's capsule has two main layers. The outermost part is an parietal layer, which is an simple squamous epithelium. This structure transitions onto the glomerular capillaries in a kind of intimate embrace to form the visceral layer of the capsule. But here is the twist. The cells in this visceral layer are not squamous anymore. They are uniquely shaped cells called podocytes. These podocytes extend finger like arms called pedicels, which cover the glomerular capillaries. These pedicels interdigitate, meaning they interlock to form an filtration slits, leaving a small gaps between the digits that acts like an biological sieve. As a blood flow through the glomerulus, about 10 to 20% of the plasma filters between these sieve-like fingers to be captured by the Bowman's capsule and funneled into the proximal convoluted tubule or PTC. And the next is the filtration membrane, your kidney's molecular gatekeeper. Let's talk about the filtration membrane, the true barrier that decides what passes from the blood into the forming urine. Where the fenestrae or a windows in the glomerular capillaries match up with the spaces between the podocyte fingers, there are only one thing separating the capillary lumen and the lumen of Bowman's capsule, their shared basement membrane. And this forms a three-part filtration membrane made up of the fenestrated glomerular endothelium, the shared basement membrane, and the filtration slits formed by the podocytes. Together, this membrane permits very rapid movement of the filtrate from the capillary to the capsule through the pores that are only 70 nanometers in diameter. Yes, that's microscopic yet high selective and efficient. Now is the tubular portion of the nephron. The tubular portion of the nephron is an direct continuation of Bowman's capsule. It is divided into three main parts. One is proximal convoluted tubule, that is PCT. Second one is loop of Henle, and the third one is distal convoluted tubule. We are now having a look into the proximal convoluted tubule or PCT. The PCT is an tightly coiled segment that begins right where the Bowman's capsule ends. It lies entirely in the renal cortex and continues downwards as the descending limb of loop of Henle. The dimension is about 14 millimeter long and 55 micrometers in diameter. The wall of the PCT is made up of single layer of cuboidal epithelial cells. A key identifying feature is the brush border. These are the tiny hair-like projection that extends into the tubule's lumen. These microvilli dramatically increases the surface area, making the PCT highly specialized for the reabsorption. Now is the loop of Henle. Deep inside your kidneys, a tiny U-shaped structures work tirelessly to concentrate your urine. This is the loop of Henle. The loop of Henle has three main parts. One is descending limb, second one is an hairpin bend, third one is an ascending limb. The descending limb has two distinct segments, the thick as a descending segment and the thin descending segment. Thick descending segment, this is a direct continuation of the proximal convoluted tubule or the PTC. It descends into the medulla, measuring about 6 millimeters long and 55 micrometers in diameter. The walls are made up of brush border cuboidal epithelial cells, which helps in the absorption. Now is the thin descending segment. This follows the thick segment and it is made up of the flattened epithelial cells without a brush border. It continues downward into the hairpin bend of the loop. Next one is then hairpin bend. The hairpin bend is also lined with an flattened epithelial cells without the brush border. From here, the loop turns upwards and continue as an ascending limb. The next one is the ascending limb. The ascending limb also has two parts, thin ascending segment and the thick ascending segment. Let's see the thin ascending segment. It is a direct continuation of the hairpin bend. It is lined by the flattened epithelial cells without a brush border. Together, the thin descending segment, hairpin bend, and the thin ascending segment measures 10 to 15 millimeters in length and 15 micrometer in diameter. Thick ascending segment. This section is about 9 millimeters long and the 30 micrometers in diameter. It is lined with an cuboidal epithelial cells without the brush border. The terminal parts of this thick ascending limb runs between the efferent and the efferent arterioles of the same nephron and forms the macula densa, a key part of the juxtaglomerular apparatus. From here, it ascends to the cortex and continues as a distal convoluted tubule or DCT. Now let's look at length and extent variations. Just let's zoom out for a minute. There are two types of nephrons. One is cortical nephrons and the second one is juxtamedullary nephrons. In the cortical nephrons, the loop is short with the hairpin bend reaching only the outer medulla. In the juxtamedullary nephrons, the loop is much longer, extending deep into the inner medulla, important for producing the concentrated urine. So, the loop of Henle's unique design, from its thick and thin segments to its precise cell types, is the kidney's secret weapons for the conserving water and maintaining the electrolyte balance. Now is the distal convoluted tubule or DCT is the direct continuation of the thick ascending limb of the loop of Henle. It lies in the cortex of the kidney and eventually continues as the collecting duct. The DCT measures about 14.5 to 15 millimeters in length and has a diameter ranging from 22 to 50 micrometers. From the microscopic perspective, it is lined by the single layer of the cuboidal epithelial cells without a brush border. These specialized epithelial cells are called intercalated cells or the eye cells, and they play a role in the acid base balance, which we will be seeing in the upcoming videos. And the next one is the collecting duct. The distal convoluted tubules empties into the initial or arched collecting duct located in the cortex as the duct travels deeper into the kidney as its lower portions lies in the medulla. Here 7 to 10 initial collecting ducts merge to form a straight collecting duct that runs through the medulla. This structure measures 20 to 22 millimeters in length with the diameter that varies between 40 to 200 micrometers. The lining consists of cuboidal or columnar epithelial cells, and there are two main cell types here, that is principal cells or the P cells, which help regulate the water and sodium balance. And the next one is the intercalated cells or I cells, which help maintain an acid base homeostasis. Now is the final part of this video, that is passage of urine from the ducts to calyces. At the inner zone of the medulla, the stride collecting ducts from each medullary pyramid merges to form the papillary ducts, also called as ducts of Bellini. These ducts open into a V-shaped region called papilla, where urine from each medullary pyramid is collected. From here, urine flows into the minor calyx. Three or four minor calyces then unite to form an major calyx. Each kidney contains about 8 minor calyces and 2 to 3 major calyces. And the final segment is urine flow, that is calyces to the bladder. From the minor calyces, urine passes into the major calyces, which opens into the renal pelvis, the widened portion of the ureter located in the renal sinuses. From the renal pelvis, urine travels down the ureter, enters the urinary bladder, and awaits excretion during the urination. And that's the journey from the Bowman's capsule to the distal convoluted tubule through the collecting ducts all the way to the urine leaving the kidney. Every structure, even cell types, is fine tuned to ensure precise fluid and an electrolyte balance. But how does your kidney know when to adjust filtration, conserve sodium, or raise a blood pressure? The secret lies in the specialized sensor right next to the glomerulus, that is, Juxtaglomerular Apparatus. In our next video, we will uncover how this microscopic structure acts as the kidney's own control tower, regulating filtration and the blood pressure with a pinpoint accuracy. If you found this explanation helpful, give it a thumbs up so more learners can find it. Share it with your friends who are studying kidney physiology, and don't forget to subscribe with the bell icon on so you never miss our next high yield lesson. Coming up next, the Juxtaglomerular Apparatus. Stay healthy, stay cheesy with Pharmacy Made Easy. Follow for more.